All right, this is James Stegan here. One practical thing off the bat. If you could, looks like everyone's muted, but if you're not talking, if you could mute yourself just to avoid background noise, etc., that would be great. Let's see. So first, thank you very much for joining us today. We're of course going to go over this WHONDRS August sampling campaign. Really excited about it. Very thankful that y'all are willing to join in the effort. It really cannot happen without you. So I just want to convey the appreciation. This is a community effort and hoping to build community resources with the community and for the community. So thank you for that. And what we're going to go over today is an overview of some of the philosophy that I already sort of started to mention there, but some of the philosophy of this effort, some of the science questions, and then we'll dig into some a little bit of the finer detail about what we're going to be doing, and have opportunity for you all to give your feedback, suggestions, any questions you want answered, and this is definitely meant to be an informal interaction. So if there's at any point you want to ask something, just go for it. One thing I'm going to do right now is put into the chat window a link to, that's a link to a Google Drive that, or Google folder, I should say, that has this slide deck. If you want to go grab that, has a couple of other things as well, such as the online metadata form, and the protocol is in there as well. And let's see, other practical thing, I'll put this on the screen. It would be very useful if you could please type your name into the chat window. And this is so we can track who has actually attended. I can kind of tell from the names in the Zoom, but not always. And it's good just to record that. Let's see, so I just said that the link to the slides, protocol, metadata form is in the chat. That's the link I just put up there. Something else, usually Amy Goldman is on these calls. This is the fourth one of these we've had. She's usually here helping me run these, monitoring the chat window. She couldn't be here today. So if there's something you want to communicate, I probably won't be able to track with the chat window. Just open or take yourself off mute and just ask a question. So other quick thing, does anyone mind if we record this? If you do, that's fine. You can send me a private chat or you can say it out loud if you have a concern about that. We'll just give people a moment in case somebody wants to say anything about that. Okay. Well, if you have a concern later for any reason, you can just email me or Amy. And if it's a big concern, we can just delete the recording. So with that, we're going to start launching here with starting with the philosophy of this. I was kind of already alluding to this, but I really want to emphasize that this effort is really, the purpose of it is to build a community resource with you, with the community, to answer science questions that none of us could answer alone. You know, we all have finite resources. We're all busy and constrained. So we're trying to take an approach to doing science that is a large collective to enable everyone. And so a key piece of that is, as we've been having these calls, we're very interested in your thoughts and suggestions. We've been making modifications. I think every call we've had so far has led to some kind of modification to what we're doing, adding a particular data type, et cetera. But we'll dig into that a little bit more later, but very open to your suggestions. Another thing is the data that we're generating from this will be immediately open after they're QC'd. So there's not like an embargo time. We're not going to hold on to the data, publish some papers, and then release it to the community. It's happening the other way around. So the data will be open. They can be used by anybody at any time for any purpose. So just as examples, if you all want to take the data and write a paper, that's great. If you want to use it to write a proposal, great. If you want to combine it with other data and do something else, that's great. we'll do our best to facilitate whatever you want to do. The data will be archived on ESS Dive. That is a DOE-funded archive. We're required by Department of Energy to put data we generate on that archive. If you go to the Google Drive that I put in the chat window, you can get this presentation, and that's a live link, or you can just Google ESS Dive. We are hoping to be generating a lot of sequence data, metagenomes, metatranscriptomes. Those will be through the Joint Genome Institute. So that's where those data will be archived. But if there's ever a question of accessing data, please just let us know, and we'll do everything we can. One of our purposes, beyond building a community resource and giving people resources that way, we are also, part of this is generating data that we need to set up hydrobiogeochemical models, reactive transport models in every site that's being sampled, to look at physical, chemical, and biological interactions, and to do this in a consistent and coordinated way across watersheds to generate more generalizable, more transferable fundamental principles and understanding to enable enhanced prediction of watershed function. So that's sort of the philosophy of where we're coming from with this. And just to give you a bit of a flavor of some of the types of science questions that we're pursuing, and I want to mention this is by no means an exhaustive list, and I certainly assume that you all have different kinds of science questions that may be of interest to you and that you might want to use the data for, and that's great. But just a little bit of a flavor. Within the US biomes, this is a logistical thing. We can ask slightly different questions in the US locations as compared to the global international locations, just due to there's certain data types we can't get globally because of just shipping logistics and things. But across the US biomes, looking at contributions of ecosystem metabolomes, and that's really, what I mean by that is detailed properties of organic matter, the contributions of those detailed properties of the chemistry relative to microbial community features, whether it be the community composition or functional trait composition or express metabolism, how those contribute to explaining variation in respiration rates of surface water and riverbed sediments across watersheds. What are the active microorganisms across biomes, across watersheds, and how do they correspond to respiration rates? Kind of related to the first question. Internationally, globally, which includes the US as well, we can't really get respiration rates globally just because of shipping logistics, but what we do still want to ask is questions about whether there's a core metabolome across rivers in terms of the surface water as well as the riverbed sediments. And of course, not everything will be everywhere. So there's a transient metabolome, and asking questions about what features of ecosystems explain variation in those transient metabolomes. Because everything's georeferenced, ecosystem features is a big umbrella. If you can look at things like watershed area, vegetation type, net primary production, sediment textures, and mineralogies. We're generating a tremendous variety of data types to help us understand what governs the composition of ecosystem metabolomes. And I should mention that I didn't put a slide in here about this, but one reason we're looking at the ecosystem metabolomes is we found repeatedly they carry tremendous information in terms of explaining variation through space and time for observed biogeochemical rates. And so far, in our experience anyways, carry more information than really anything else, more than microbial communities or any kind of bulk geochemistry or anything like that. So we're very interested in understanding more about them for fundamental reasons, but also to build enhanced predictive capacity. And this last bullet, I kind of already alluded to this, but I just want to reiterate that there's lots of other science questions that we've thought of, and I'm sure you will as well. And we certainly encourage you to contribute your ideas, pursue your own ideas with these data, etc. So it's very, very open that way. One thing, so this is a map of an approximate map. This isn't precise, of the domestic locations in the US that will be sampled with this effort. And the point of showing this is not to, to dig in any particular location, but rather just to emphasize that we are covering a lot of different locations across a lot of different biomes. A lot of this is enabled, not all of it, but a lot of it is enabled by existing infrastructure funded by NSF as well as DOE, US Forest Service, some USGS, but also enabled by individual investigator field sites. And we very much appreciate everyone's efforts and willingness across the board there. What I'm not plotting here are international sites. Some of these are still being worked out. I think Eric is on the phone. There may be other international folks. Eric Batos is on the phone. He'll be doing some Canadian sampling. So I didn't put those locations here, but we are covering some locations across Canada, Europe, and Israel. And I already kind of mentioned that that does include a subset of the data. In particular, as I mentioned, we can't do respiration rates internationally. But one thing also to mention is that this is a Northern Hemisphere effort, this go around. And the reason is we wanted to constrain the time period to control the season, basically, across all the samples that are being generated. So we haven't committed to, but we have been talking about having a Southern Hemisphere effort 6 months later. So the season is approximately the same, even though it's 6 months separated in time. And that timing of sampling is quite important. We want all the samples to be taken in a short time window. We're not going to try to do this on the same day. We couldn't even handle it if that happened. But we want to look at systems that are getting relatively close to being in base flow. Recognizing that's not the case across all the systems. There's lots of variability. We wanted to constrain the time period here. And so that's very important in terms of when you all go to the field. We're hoping that occurs within this August 5th to August 20th time window. That is not super strict. There are a couple of people that are already falling a little bit outside that window. But if at all possible, we'd like to get people to stay within that window. And we can certainly work with you if there's concerns or issues with that. And we know, I mean, you can only go to the field when you can go. And so we're trying to be very flexible. The fourth bullet here, super critical. All the sites that are being sampled, they do need to have a stream gauge. That could just be a pressure sensor. Doesn't have to be discharge per se. But that data does need to be shareable, as I already mentioned. All the data are public here. We want to be able to share those data and would like the data to be collected at least every 2 hours, so we have some good temporal resolution. Ideally, with at least 6 months of pre-sampling stream gauge data. That's we don't have to go all the way to 6 months. A year would be great. 6 months, good. A couple months, probably acceptable, but not quite as good. And so if there's concerns about that, we're very happy to work with you, find alternative solutions, etc. So again, we're trying to be as flexible as possible, but hold the line to make sure we have those contextual hydrologic data. So I want to pause for a moment and just open it up if there's any. We're going to go through a bunch of other stuff, but are there any questions, concerns, etc.? We're happy to dig into whatever you like at the moment.
Mm-hmm.
Okay.
Hi, this is Jenny Dugan. I came in a little bit late, but I don't have a stream gauge. I'm working in an intertidal area, so I just, relative to that last bullet.
Okay.
We can maybe have an offline conversation about that, but I just wanted to put a little marker in.
Okay. Yeah, let's have offline discussion on that and see what kind of creative solutions we can come up with.
Yeah, okay, great.
And also for Jenny or others that have joined more recently, I will put I'll put a link again in the chat window here. That is a link to a Google folder that has this slide deck and some other materials. And also if you could put your name into the chat window, just so we can keep track of who has been on the calls and who has not, that would be very useful. Thank you. Anything else at the moment? Okay, well, if that's something that comes up later, please let me know. So we're going to start to dig into a little more detail here now. So this is an overview of the types of data that we're generating. And we have water collection, we have sediment collection, and leaf collection. Let's start with the water. So basically, there's a variety of different vials, some filtered, some unfiltered. We'll talk a little bit more about this on the next slide. We are preserving the filters themselves with RNA later for microbial analysis. And one thing I want to mention here at the top, isotopes. This is specifically doing water isotopes. And this is a nice example where talking to the community, this was suggested to us by Steve Good at Oregon State. His perspective was, if you're going to be out there collecting water samples, it's pretty easy to get a few more mils of filtered water and do water isotopes. And he often uses that data type to look at evapotranspiration aspects. And that's something that we're not doing directly, but that's a simple modification to the protocol. Doesn't take much time. It's a simple analysis to do. And so we added that in here. And more than likely, he'll probably take those data and try to do some analyses beyond anything that we will do. We're very open to those sorts of suggestions. We can't completely change our protocols and add all kinds of other new things, but little things are very much of interest to us. In addition, doing carbon concentration, we're doing FTICR mass spectrometry. That's, if you're not familiar with it, is a method that's a very high resolution approach to do untargeted characterization of ecosystem metabolome assemblages, basically characterizing organic matter in the environment. We're also, in addition to that, also to characterize organic carbon, we're doing EAMS. And that's a nice example of a different kind of an approach where we don't have much EAMS expertise internally here. So what we're doing is when you guys send your samples, we're collecting all the vials for EAMS, and then we're going to ship all of those up to Rachel Gabor at Ohio State. She has tremendous expertise in that method. And so that's where, we talked to her at a conference and she was interested and we worked it out. And so that's a set of analyses that's outside of PNNL, but it's still a part of the community effort. And those data as well will be available to everyone. So just emphasizing, we can think about different ways of generating data together. Let's see, beyond that, we'll be doing incubations of that water, looking at oxygen consumption, enzyme assays that's another one that was suggested to us and will likely happen outside of PNNL. In fact, that one the person that's interested in that may be writing an NSF proposal to either use those data in their proposal or use the proposal to generate those data. We'll see. Microbial biomass, that one we're actually shipping samples off to Israel for them to do this via flow cytometry. Another nice example of doing analyses with the community. We're doing anions, cations, specific conductivity. Down here is listed all the specific analytes that we're doing. I already kind of briefly mentioned the microbial work. This is being done through the Joint Genome Institute. We have a proposal in review there with Kelly Wright as the lead PI. There's about 10 PIs on that proposal. This WHONDRS effort is only one piece of that proposal. It's a proposal to do microbial sequencing across tons of rivers all over the world, even way beyond what we're doing here. On the sediment side, we are doing also the same kinds of incubations as is happening with the water, looking at oxygen consumption. Same FTICR, bulk carbon and nitrogen, grain size, mineralogy, same enzymes. For the sediments, we're also adding NMR as an approach. If you don't familiar with it, it's an approach that is good at quantifying small metabolites, and it complements the FTICR quite nicely that way. FTICR is not quantitative and it targets larger molecules. We're not doing it on the water, principally because usually if you just collect surface water, stream water, the concentration of small molecules is much too low for the NMR to see. but from the sediments, we think we can make this work. And then additional sequencing from the sediments, as well as doing qPCR of the 16S gene to get another sort of proxy of sorts of microbial sort of cell density, in addition to flow cytometry. Let's see, I guess that's not actually listed. There's microbial biomass over here on the sediment side via flow cytometry. So in the leaf sampling, this is something we're sort of beta testing. What we're likely to do with this, well, basically what it is collecting just 10 leaves from just the single most dominant riparian vegetation species that's adjacent to where you're going to do your sampling. So this isn't, you don't need to do a quantitative analysis to know which one's the dominant. You don't need to look at the whole watershed. It's just visually, usually you can tell, okay, there's a dominant riparian species, and just pick 10 live leaves. We'll send you an envelope for that. Once they get here, we'll dry them and leach them to look at concentrations, what comes off, and also characterized by FTICR. And the purpose of doing this really is just sort of, again, kind of a beta test of trying to think about using those leachates to do laboratory experiments with a common set of sediments. And so we're, one can maintain the sediments and the microbial communities, but vary the carbon profiles that are coming from vegetation. One thing I'll mention is that we know we're not going to get leaves from all the sites. For example, I think it's about 27 of the sites are from the National Ecological Observatory Network, or NEON. They are not permitted to collect leaves, and so we're just not going to get leaves from those sites. If you have any issues in your site that you're not allowed to collect leaves, that's fine. Don't worry about it. Just skip that piece. So I want to pause here for a moment, get people's thoughts, suggestions, questions, any other tweaks you can see that might be beneficial, in particular beneficial to you, things you might be interested in. So, thoughts? Okay. Well, if anything else comes up, springs to your mind later, just feel free to speak up. Okay, so this next slide is kind of a high-level overview of how the sampling works. And first, when you get there, what you want to do is identify 3 sampling locations. And so, as I mentioned, we need to be near a stream gauge. And what we're doing is sampling in 3 locations relatively close to that stream gauge for the sediments. And by relatively close, I mean, hopefully within about 100 meters of that stream gauge. And really what matters is that, does the stream gauge reflect the hydrology at the locations that are being sampled? If you can say confidently, like, yes, that's good, then you're close enough. This is sort of the litmus test there. So basically figure out where you're going to sample. And I should mention, this is in the protocol, but we do want to be sampling in depositional zones where fine material is likely to settle out. When we get the sediment, we will sieve it to less than 2 millimeter. So we are focusing on that smaller size fraction. And also, one of the reasons for that, there's many reasons, but one of them is we're trying to follow sediment sampling protocols that NEON has laid out. And so that the data that are generated here are as close as possible to the same protocols that NEON uses, so the data are as comparable as possible. So identify those locations. We have some drawings in the protocol about what a depositional zone is. If you have any questions, happy to discuss that. There's some metadata to record. We'll go over that. It should be pretty quick. There's some pictures to take of the sites to give us a sense of what's going on, looking at sediment textures and things. That's all detailed in the protocols, in the protocol document. Measure water temperature, pH. We'll send you a pH strip. It's not super precise, but just kind of gives a ballpark. If you have a pH sensor, great. And dissolved oxygen here, it says optional. If you have the ability to measure oxygen, that's great. Like if you have a handheld YSI or maybe a sonde or something, that is fantastic, but we recognize not everyone has that instrumentation, so that's why it's optional. And just really trying to get some of the basics down here. So then once you get where you know where you're going to be, and you've written down a few bits of metadata, the next thing to do is to collect the water. And we're getting water from just one, as it says here, just one of the sites, specifically the most downstream sediment sampling location. So first, you'll get some unfiltered water, some filtered water. Once you've filtered the water, you'll need to push you will need to push some additional water through the filter, and then preserve it. And the reason to push more water through it is we have to filter enough water to get enough microbial biomass to actually be able to do metagenomics and metatranscriptomics. And so that's detailed in the protocol. It shouldn't take long to filter the additional water. If the filter clogs, then you're good and done. And related to clogging filters, which in some systems will be rapid, other systems won't clog at all. Something that's important, and this is in the protocol, we have the water samples we're collecting, as you saw in the previous slide. There's a number of different kinds of analyses that we're doing, which means different kinds of vials, different preservation methods. We are collecting replicates of different data types, different vial types. And so what we would ask is, if clogging seems like it's going to be an issue in your site, it would be good to try to get at least one replicate from each vial type, each sample type, before you try to get all replicates for a given sample type. Just so, if you get all replicates for one sample type and then filters clogged and you can't get replicates for other data types, not so ideal. Better to at least have one replicate from each vial type. Happy to discuss that more if that's not clear. So you got your filter preserved. That's in RNA later. You're not freezing anything. We'll mention that again a couple times. Actually, that's really important. So after that, that's when you move to the sediment collection. And really, it's the 3 sites. Do each site in sequence, starting downstream and moving your way upstream. Sediments are basically going into a 125 milliliter jar. These are surficial sediments, 1 to 3 centimeters. Again, that's what NEON does, so that's what we're doing here. One caveat is, systems that have fine sediments, sandy or silty, muddy, be very easy and quick. Other sites may have an armored riverbed. That's what we have here along the Columbia River. Lots of cobbles. Getting that finer grain size fraction can be a little bit challenging. And what is totally acceptable is, if you're in that situation, start moving some rocks around and you can dig a little deeper to get into finer sediment that may be in the interstitial spaces among the cobbles. Just make a note of it. If you have to dig down 10 centimeters or 20 centimeters, that's fine. Just make a note, please. You'll see in the protocol, we ask that to remove any large material, anything basically over 4 millimeters. It'd be great. Just picking it out, basically. You won't be doing any sieving. We want to keep this as streamlined and as quick for you in the field as we possibly can. At the same time, you'll be parsing some of those sediments into a Falcon tube, 50 mil, actually 2 50 mil Falcon tubes that will have RNA later in them already. And you'll need to fill those to a particular level of volume of sediment so we can get the right ratio between RNA later and sediment. And that's all detailed in the protocol document. And let's see. So then once you have the sediment done, if you can, collect those leaves from the dominant species. Should be quite quick. And then the completion step is really to refrigerate samples. As I already mentioned, don't freeze anything. The only thing you want to freeze is the blue ice packs. We'll send you the blue ice packs. Just put them in the freezer. That's what's going to keep the samples cold when you ship them back overnight. And we're paying for all the shipping. You'll ship back in the same box that we send stuff in. And we'll actually, there's a slide just on shipping that comes next, so we can dig into that more. But refrigerate, ship the next day. I guess that's not detailed in this particular slide, but basically you sample, stuff goes in the refrigerator, and ship the next morning overnight back to us. We're trying to minimize the time between sampling and our sample processing and the incubations that we'll be doing. Also very important, please let us know when you've sampled and when you've shipped. As you saw on that map, we're covering a lot of sites, and we really have to know when samples are coming in. There's a lot of logistics on the laboratory side that our team needs to know how to work through. Also, the online metadata form is super important. It parallels the hard copy metadata form, and which we'll go over, and I'll show you the online form. The online form is in that Google folder that I set a link to. You can go look at it if you like. So let's see. I'm going to just pause again for a moment. Do people have any burning questions, or any questions really about anything so far, or suggestions? Okay. Let me know if anything crops up. Okay, so let's look at this. This is, you'll get a hard copy of this in your sampling kit, and this is the metadata form. It looks like a lot, but it's really not. This should only take a few minutes to fill out. So it's a lot of basic stuff. Your name, your organization, the kit ID, that'll be obvious when we send it to you. If you have a different site name or ID that you use for your site, please put it in there. The date, the time that you're sampling, the start time. Please include the time zone, just so we know that's quite critical. Name a river. This stuff down here, you're just making some visual, very quick visual interpretations of the environment. So you can pick 2 of these to give a sense of the vegetation. Do you have some broadleaf? Are you in a grass system, a cropping system, not vegetated? Just pick up to 2 of those. General geomorphology. Are we talking about a single straight channel system, a multi-channel, a meandering system? The gradient, is it steep? Are we like in the mountains and we're in a steep place, or a relatively flat lowland area? Very qualitative stuff here. Similarly, dominant sediment type for just the superficial sediments. Just give us a general sense of what we're looking at. If it's a super muddy place or it's an armored cobbly place. That's all that. In the middle column here, as I mentioned, there's 3 sediment sampling locations. And we'd like to know how far apart those are from each other. Just to give us a sense, if they're 5 meters apart from each other or 50 meters apart from each other, we just want to get a ballpark on this. So if you've got a field tape that you can use, and if your sites are close enough that makes sense, take a real measurement, that's fine. If you don't have that, or it doesn't make sense for any reason, it's totally fine to just pace it off. Just give us a rough idea of the layout among your sites. Coordinates in decimal degrees, please, for each the upstream, midstream, and downstream sites. This is totally fine if you don't have a GPS. Most people, not all, but most people have a smartphone. You can just get an app to get your decimal degree coordinates off your phone. Very, very simple. Doesn't need to be wickedly accurate, just giving us a sense of where we are. Quick measurement of the height of the water column will send you a measuring tape to do that. And that's really most of this stuff. Here's the pH. Again, that's just a pH strip. Temperature, you will have to provide your own thermometer. We can't send thermometers to everybody. Most people have a thermometer though, so it should be simple. DO, sorry, my screen's getting blocked by that little thing, but the DO, if you have a way to measure that, great. Here is giving us again, a very rough qualitative sense of the kind of depositional zone that each of your sites is. If we're talking about a riffle, or are you downstream from a big log, or just something shallow along the shore, this isn't something you want to be spending more than 30 seconds evaluating. Just a quick idea what we're doing. And all you're doing is putting letter designations. You can see U, M, or D in these boxes. These are all suggestions here from the community during I think our first or second call we had, of giving us a sense of, are there lots of algal mats, high, low, or no, each of these sites? Do they have a lot of submerged macrophytes, plants growing in the water, high, low, or no, just for your different sediment locations? are you in a system that has a closed canopy or a partial canopy, or is it wide open? Just quick visual inspection, just give us your best ballpark on that. As I mentioned, there's pictures to take. That's all detailed in the protocol. Any other sampling notes? What's the weather like? Anything that deviated from the protocol? Did you have to dig a little bit deeper to get to those finer sediments, et cetera? Anything goes there. Any questions, concerns, suggestions on this piece? Okay. All right, moving on. So this is shipping logistics. I already mentioned this is very critical. So before we actually can send you supplies, we do need to know what date you intend to sample. We've been working with people. They had 2 potential dates, and we helped decide which date to go for. We do need to know that before we can send you stuff. And also so that we can include the right FedEx information for getting the stuff to you, but also getting it back to us. So we're paying for the shipping in both directions. We'll provide you a shipping label to get it back to us. So it should be pretty streamlined. I kind of already mentioned this, but you really do need to plan to ship the morning after you sample. We can't be holding samples for periods of time. This is also really important, if at all possible. We know it's not always possible, but if at all possible, it would be great if you could sample on a Sunday or a Monday, thereby shipping on a Monday or Tuesday. We do have capacity and an ability to deal with other situations. We know they exist. Not everyone can go on those days, but we have to process the samples. We have to start the incubations, and samples showing up later in the week push our team into weekend work, and the logistics are becoming increasingly difficult for stuff showing up later in the week. But if it's a problem, we'll work with you. We'll figure it out. Just please communicate with us. Packaging, you'll use the same box you receive all the stuff in. That's what you'll ship samples back in. Pack it as close as possible to the shipping time, so it's not just sitting around in the lab warming up. If you're going to ship in the morning, then pack it that morning, put the blue ice in there, and ship it away. And I already said this, but I'll just continue to reiterate that the blue ice is the only thing you want to freeze. Don't freeze any samples. Samples just stay in the refrigerator. And we do have one situation where the collaborators don't have access to a refrigerator. They're just going to be camping in the field. So what they're going to do is just be holding samples on wet ice and then shipping with the blue ice. So we'll work with you and find creative solutions. So this comes overnight for US locations by FedEx. That's, again, that'll be all paid for, and you get the shipping label and everything. So there shouldn't be anything you need to pay for. This last bullet is, I already said this, make sure you let us know when you're shipping the package. And please use this email address, thewonders@pnnl.gov. If you send an email to me, and it's just to me, I am not great with email. I'm getting hundreds of emails a day. Chance I'll miss it. But if you send it to that email, it will be seen. So please do that. Okay, this is the last slide. And this is really, again, you can go get this slide deck, and these are live links. There's some various contact information. There's some videos, WHONDRS videos on YouTube, link to the protocol. And what I want to do now is, one, thank you again. WHONDRS really cannot exist without you all. It really is a community effort. And I really have appreciated the suggestions we've gotten from the community. We've taken all of them quite seriously and made a number of modifications to what we're doing. And so at this point, I'd like to open it up. If you have any questions, any concerns, any ideas, etc., everything is open to discuss at this point. And if there's anything you want to discuss later, we can also do that offline. That's completely up to you. So, any thoughts?